Okay, processing eukaryotic mRNA. The first thing for you to note is that we've got the word eukaryotic here. Eukaryotes are different than prokaryotes. In something that is a eukaryote, that means that the DNA is found inside of the nucleus. And if we have something that is a prokaryote, I always think prokaryote is primitive. A prokaryote has its DNA that's wound in a circle and it's kind of free-flowing throughout the cell. So if you think like a bacteria doesn't have a nuclear membrane, so the DNA is just kind of wound up tightly inside of the middle of the DNA. But in eukaryotic, which would be your plants, your animals, your fungi, and then some of the protists, we've got these genes, and we're going to be looking at how the mRNA is used to read these genes. So what we have in a gene is we have, or a piece of DNA, is we have what's called an exon, which is a coding region, and we have an intron, which is a non-coding region. So exon, this coding region, the coding region is where we actually come up with the amino acid code to make the proteins. So the exon is the important part in being able to make proteins. The intron is called, sometimes it's called junk DNA. It's something that doesn't actually do anything that we recognize in the DNA transcribing process. And so it doesn't make any code. It doesn't do anything that we know of. Someday it will probably come out that the introns have some really important part in making us who we are or leading back in our evolutionary past. But for right now, scientists don't know exactly what that does. And so what we're seeing is that during transcription, we have what's called a 5' cap and a 3' A tail. So what's happening here is in our primary piece of RNA transcript, so we've got these regions where we've got transcribing from the piece of DNA to the RNA. We're getting all of our information in order. And then the next step is that we take all of those introns and cut them out and put all of the exons back together. So that mature piece of mRNA transcript is packed tightly with things that actually matter. If it was all just these trillions or billions of base pairs of stuff that did stuff, and which is the exons, and stuff that doesn't do stuff, the junk, the introns, it would be way too long to actually make pieces of protein with. So they take out, the introns are cut out by the cell, the coding regions are put together and spliced together, and they come together in this 5' cap, which is where it starts, and the 3' tail, which is where it ends, and we've got one long piece of mature mRNA that's ready to start reading and making amino acids. So the basic process, if you think of this like your grandmother's cookbook, is that inside of the nucleus of the cell, we've got our DNA, which has genes that are coded into the DNA. So the idea of your grandmother's cookbook, all of those recipes are inside of one place. So all of that DNA is inside of the nucleus. And then the next step is transcription. The sequence for a gene is copied from DNA to a middleman molecule called mRNA. So we're going from the DNA to the RNA. So this would be like if you had your grandmother's cookbook and you got out an index card and you started copying one of the recipes. Say you didn't want your entire grandmother's cookbook, you just wanted 1 or 2 recipes. So you take an index card and you start writing down that recipe from your grandmother's cookbook. Then the mRNA has to get out of the nucleus in order to get into the cytoplasm and go out and pick up those amino acids. So that would be if you were to take your index card with your recipe and take it out of the kitchen. It's basically getting the grandmother's cookbook to the index card and then taking it out of the kitchen to your kitchen. So our mRNA there. Then translation, the sequence for a gene is now encoded in mRNA. It's used to direct the production of the protein. So this would be if you took your index card and you got out all of the supplies in order to combine this together and begin to think about making your recipe. So combining all of those baking supplies together, you can see there the strand of mRNA and those little circular dots there at the top. Those are the amino acids that are being coded for in the mRNA, and they're being put together. And so then the final step of translation is that we have this protein molecule. Now this looks deceptively simple. Imagine that this is a much bigger process, and also that it's happening in 3D. We've got these amino acids, whereas they just look like little circles here. They're actually these really complex, different shaped molecules, and they fit together in this protein that is this really complex 3D model. Just like your chocolate chips aren't just flat pictures of chocolate chips. They actually have depth and shape and substance to them. The amino acids that are built into the protein have that same amount of substance to them. So one of the things that I want to point out to you as you're learning about this process is to think about what the words mean. Thinking about transcription and translation, I know I always have a problem thinking, well, which one comes first if you're trying to remember it on a test? Which one comes first, the transcription or the translation? So let's think about this if we were translating something from a foreign language. Imagine if I went and I heard somebody singing a song in Spanish. The first thing that I would want to do is I would want to get the lyrics to the song. So I am transcribing the song. I'm going from words to the lyrics on a page. And then I would take those words on a page over to a translator and say, can you translate this for me? And then they'd give me the output, which is the English version of the song. So the first thing you need to do is write down the words. The second thing you need to do is find out what the words mean. So that's how genes work. Let me stop this.